We have a worrying future in front of us. In 2050, there will be 10 billion people, and we will have to produce about 60% more food than at present.
Periods of severe drought, sudden temperature changes, an increasing number of people but less arable land.
As scientists, we must find technologies and ways to grow our cereals, our plants, also in marginal environments. Plants, like humans, are meta-organisms that live together with many microorganisms. The 2 millimeters of soil near the root, called the rhizosphere, is their intestine. In this zone, the plant exudes to recruit and maintain its microbiome.
That is, those microorganisms that help and increase the health of a plant, and that allow us to reach higher yields of food production. The microbiome of the plant is divided into different ecological niches. The microbiome in the root, the microbiome of the green part of the plant, that is, the leaves, the fruits, the stem, and the microbiome of the soil. That is precisely the reserve from which the plant recruits the good microorganisms, those that have a beneficial function for the plant itself.
Research into the microbiome exploded about 10 years ago. Beforehand, it was impossible even to think of undertaking this, because we knew that there were many very complex communities, but we couldn't study them. So what's happened in recent years? We've been able to sequence DNA very quickly, and it's very economical.
We study microbiomes through the use of DNA sequencing, and especially through the identification of gene markers that give us the name of the bacterium and that tell us which bacteria are present in a given situation and in what quantity.
With this information, we can understand who the main actors are, the bacteria that are at the heart of these communities and that have the most important role in these communities.
Isolate them, characterize them, and use them in the form of bioinoculants to give to the plant.
There are also microorganisms that we can use to control diseases,
in order to replace chemical fertilizers that are still being used.
One of the methods we are studying together with companies is seed coating.
Coating seeds through the use of biofilms of bacteria.
Alternatively, these can be put directly in the soil before planting the seed.
The advantages of bioinoculants over chemical fertilizers are, above all, at the level of the environmental impact they have. We are not introducing any microorganism that was not already present in nature, but we are simply giving a greater bacterial charge of microorganisms to the plant that has a beneficial effect on the production and health of the plant itself. Moreover, the amount of available chemical fertilizers continues to decrease and costs more. Developing countries, for example those in Africa, have very little access to chemical fertilizers. Therefore, the use of organic fertilizers that will cost less than chemical fertilizers will be of great help.
The ICGEB is a unique organisation that has 3 research components.
The headquarters in Trieste and components in Cape Town and New Delhi. It's formed by 66 member countries.
We work together with these member countries. We have scientific networks because our mandate is
to promote science and research in developing countries.
Here at ICGEB, we have as our objective different scientific themes that align us perfectly with SDG 2 of the United Nations for Zero World Hunger, because we are studying many mechanisms that plants can use to be more resistant to pathogens and more capable of growing in extreme conditions. Different technologies, different scientists will lead to plant varieties that will be much stronger, and also to products based on microbiology that will allow us to increase sustainable agriculture in the future. So attaining 60% more food by 2050 for 10 million people is a very important challenge, and it's also worrying. But as a scientist, I'm very optimistic, because I see many significant technologies, scientists who are very determined, and also scientific collaborations that will surely enable us to reach this great goal.
Defeating hunger in the world is possible thanks to the tools that we have, and the studies that we are bringing forward, thanks to which we will make land more productive and able to feed greater numbers of people.
Sempre più maggiore di persone.